All right. So welcome again, everyone. My name is Bridger Layton. I am the Education Programs Coordinator for the Methow Conservancy. And I'm really excited to see some familiar people, but also I think some folks from a new audience. We're excited to partner with Joshua at the Wenatchee River Institute for this event and excited that we get to kind of cross-pollinate a little bit. So perhaps for some of you, this will be the first interaction you have with the Methow Conservancy, and welcome to that community. I do want to shout out a few fun things that we have coming up. First off, on March 14th, we have a talk scheduled at the Twist River Tap House with Home Range Wildlife and Conservation Northwest, and they'll be talking about the intersection of wildlife, conservation, and recreation in the North Cascades. Next month's first Tuesday will be in person in Winthrop at the barn, and we'll be learning about the Karelian Bear Dog program from bear and cougar specialist Rich Beausoleil, who works for WDFW. And then in May, we're back on Zoom again, partnering again with the Wenatchee River Institute, and we'll be learning about how native plants in the Cascades are responding to climate change. And then the last thing I want to mention is that the Conservancy are in the midst of a really exciting project called the Campaign for Sunny M Ranch. Many of you will already be familiar with our ongoing effort to preserve wildlife habitat, protect recreation access near the town of Winthrop, and also create affordable housing for those who live and work in the Methow Valley. This project taps into so many of our values as an organization, and it'll shape the future of the Methow in important ways for locals and visitors alike. You can learn more about that project on our website or at an upcoming Zoom info session, which if you go to our events page, all of the info you need is there. And then for our Wenatchee audience, we're actually hosting an in-person session about the Sunny M project in Wenatchee on March 22nd. If anyone wants to, come ask us questions in person. If you're new to our audience, please join our mailing list. We have 2 newsletters each month. One is a naturalist bulletin that I write that I have a lot of fun with, and the other is our e-news, which is incredibly informative and I think very well written. My sense is that people actually like getting emails from us. I'm biased, but I hope you'll hop on the list. So with that, I'm going to hand over to Joshua at Wenatchee River Institute, and he's just going to give a quick hello as well before we dive into the evening's program.
Thanks, Bridger. Thank you for letting us co-host this. And Wenatchee River Institute, we're located in the Wenatchee Valley, and we're a nonprofit that our mission is to connect people, communities, and the natural world. So when you reached out, Bridger, for this, I thought this is a great fit for what our audience and our community would love to hear about. So I just want to thank you again for letting us hear all this.
So to start our formal program for the evening, I'm going to go ahead and show a quick video clip that is an overview of the Predator Prey project, and then I'll introduce our speakers for the night, and we'll be off and rolling. Assuming I can make screen sharing work.
So fundamentally, the Predator Prey project is trying to answer what seems to be a relatively straightforward question, which is, what are the impacts of wolves and predation in general on various ungulate populations in landscapes with people? But there's a lot more that goes into that than just, a simple who's eating who sort of thing.
The Washington Predator-Prey Project was started as a legislative mandate in 2016. Our agency was asked to conduct research looking at the effects of wolf predation on the ungulate populations in Washington state. And we're working together with the University of Washington. They're looking at it.
Serving in the legislature, you're only as good as the information that you can get. You know, we've had questions over the years, you know, how are our ungulates doing? You know, what are the populations of moose, elk, deer? People are interested in that in this part of the country. And then we've got some new predators moving in. Well, let's get some good science on it.
Initially, it was just sort of, what are the impacts of wolves? And we said, okay, well, we can answer that. But if you really want to know, you can't just leave it at wolves.
That's a cat.
You have to understand what other carnivores are doing to ungulate populations, and you have to understand how those ungulate populations are competing with one another and how those carnivore populations are competing with one another. And then to make things extra special complicated, you have to incorporate that human element.
Wolves in Washington are recolonizing in working landscapes. They're not recolonizing these pristine park-like settings. They're recolonizing areas where people live, where they work, and where they recreate. And so we know that we can't take the lessons learned from Yellowstone and apply them here in Washington. We know it's going to be different.
With the Predator-Prey Project, there are 2 study areas.
In the Northeast study area, we've got a wolf population that's well established. Where in the Okanagan, they're on what we call the colonizing front. We can then answer questions not just of who's eating who and where and how often, but what's the impact when there's fewer wolves versus more wolves.
All right, so hopefully that provides a nice overview, and one of our board members actually helped make that video, which is an exciting thing, Benj Drummond and his partner. So I'm going to go ahead and introduce our speakers for the night, and they'll share more about the project through each of their research lenses. First, we have Dr. Sarah Bassing, who is a research scientist at the University of Idaho. She studies species interactions, human impacts on wildlife populations, and species distributions with the goal of informing wildlife conservation decisions. We have Dr. Taylor Ganz, research scientist at the University of Washington, who studies how ungulate populations are influenced by predators, forage dynamics, and large-scale landscape change, including wildfire, timber harvest, and climate change. And then Lauren Satterfield, PhD candidate at the University of Washington, who is investigating wolf-cougar interactions across anthropogenic landscapes in the Pacific Northwest. And finally, Dr. Laura Prugh, who is an associate professor of wildlife sciences at the University of Washington. And Dr. Prugh's research examines wildlife community ecology in the context of a changing world. She's studied carnivore community dynamics for the past 25 years through intensive field studies in Alaska and Washington. I know that's super brief. If you're interested in learning more about each of these folks, we're going to go ahead and put more detailed information about each of their careers and the projects they're working on our website. If you go to the archived events page and click on this event, that's where you can go to find the recording link if you want to share it with others, but also a good way to learn more about these folks. But we wanted to keep it brief in order to give them the time they deserve. And so with that, I'm going to go ahead and hand it over to Dr. Sarah Bassing.
Great, thank you so much. All right, I will share my screen. Okay, so everyone should be seeing a picture of a deer. Awesome. Well, I think I can speak for everyone here on the Predator-Prey Project and saying that we're very excited to be here and get to share some of our research, especially since a lot of this work came from the Okanagan, Methow Valley. So we're really excited about this. So my research is obviously part of the Predator-Prey Project, and I focused on spatial and temporal patterns of predator-prey interactions across a human-dominated landscape. So, there we go. So generally when we think about predator-prey interactions, we think about the fact that predators kill and eat prey. It's kind of straightforward. But in reality, these interactions are much more complex than that. And this is in part because prey have a number of anti-predator behaviors that they're going to engage in to help them reduce risk from predators, whether that's avoiding risk or keeping an eye out for predators. So these can influence those predator-prey interactions. In addition, predators have a number of behaviors of their own that they can use to increase the success of locating and then capturing and killing prey. And so because of that, we know that predation risk can vary across space and across time, whether that's over the course of the day, the season, the year. So there's a lot of variation in risk to these ungulate species that we have out in eastern Washington. And then this gets more complicated when we recognize that, and particularly in Washington, but across the globe, we often have these multiple predators and multiple prey species all interacting together. So this can create a very complex picture when we're thinking about predator-prey interactions. So my focus on this predator-prey project out in Washington was to really think about this broader community dynamic where we have all these species interacting together, and to think about how these multiple predators and multiple prey species interact, and what kind of patterns arise out of those interactions. And specifically thinking about space use patterns like habitat use or movement of these species, as well as activity patterns, such as when are animals most active across the time of day, and how interactions between these different species influence those patterns. And then on top of that, my research also incorporated trying to figure out how humans factor into those relationships. So to do that, I used camera traps, which are a pretty nice ideal tool used for monitoring multiple species at once. So this plot here, this map shows those two study areas that were shown in the video. The different colored dots represent our camera locations over the course of 3 years of data collection. And these cameras allow us to get information on multiple different species using the same device, which is really handy. And so the species that I focused on for my research included mule deer, elk, and white-tailed deer, which were the primary focal species for the predator-prey project, but also I looked at moose. And then I focused on 5 different predator species: wolves, cougars, bobcats, coyotes, as well as black bears. So you'll be hearing a lot about these species over the course of the night. So to get these pictures of these different species, we deployed cameras. We put them across both public and private lands. So we've got a broad sample of the different type of habitats these animals are using. Of course, we're on private property with permission of those landowners. And we put those cameras on trees, fence posts, or shrubs, something that would allow us to prop the cameras in a way so that they could monitor linear features such as unpaved roads or hiking trails or game trails, areas that would funnel animal movement. So we had a better chance of detecting all these different species. Now, because we were trying to get images of animals as small as a bobcat and as large as a moose, that meant that we had to angle the cameras just right and make sure that they were actually triggering on animals of those different sizes, which meant that we ended up doing a lot of carnivore crawls. We ended up crawling on the ground a lot to make sure that these cameras were angled just right and we're going to snap pictures of all these animals. So we ended up getting pretty dirty, but this allowed us to get a phenomenal amount of data. We ended up with thousands of pictures of these different species. So using those data, those different images of all these different wildlife, I used those images to answer a number of different questions for my dissertation. And one of those questions was really focused on how humans influence the spatial and temporal overlap of predators and prey. So that's what I'm going to focus on sharing with you tonight. So generally, when we think about predator-prey interactions, we expect predators to use areas that have prey, where those prey are. That's obviously a prerequisite for predator-prey interactions. Predators need to be using the same places as their prey if they're going to locate and then capture prey. So we considered this spatial overlap, where we had both predators and prey using the same general areas. We also expected that predators were going to be active in areas where, or at times when prey were also active. Because again, if prey are active, predators have a better chance of finding that prey and then capturing them. So we consider this temporal overlap, the times of day or night when both predators and prey were active together. But when we bring humans into the picture, that might change those relationships and when and where these predators and prey species are active together. So this might arise changes in their relationships due to a number of factors. Depending on the species and the human activity or land use, we could see different species avoiding human activity or being attracted to human activity, all of which could change the spatial or temporal overlap between these different species. Now, out in eastern Washington, 2 very common forms of human activity and land use that we were interested in exploring were livestock grazing during the summer and hunting activity during the fall. So both of these take place across both public and private properties, and give us a lot of human activity and ranges of human activity that could influence both predator and prey overlap, as well as ultimately influencing predation risk. So to explore this question, we analyzed those camera data with respect to a number of different factors that we thought related to livestock grazing and hunting activity. So specifically, we looked at cattle activity, as well as whether the cameras were on public grazing allotments or not. Specifically, this was during the summer season when livestock are generally turned out on public grazing allotments. And just to be clear, when I talk about cattle activity, I'm specifically referring to just the number of pictures of cows that we got on camera. So we were not looking at unique individuals. We weren't looking at composition of these herds or counts of individuals. It was just, were there cows there, yes or no, and then how many pictures were there. Similarly, we looked at hunter activity as well as whether cameras are on public versus private property, because there should be variation in the amount of hunting activity on those different types of land ownership. And we were specifically looking at whether or not hunting activity or human activity, or land use changed those daily activity patterns or habitat use for these different species, and then whether that resulted in changes in predator-prey overlap across space or across time. So for example, looking at these two pictures of the hunter and the deer, we might expect that if deer are going to become more active at night to avoid human hunters during the daytime, then maybe that could also increase the times of night when deer and their nocturnal predators, such as cougars, are both active, and thus changing the amount of time these animals are overlapping over the day together. So I'm going to skip over all the gory statistics to get at those analyses and just report some of the cool results that we've got. So first of all, in general, we found that livestock activity and cattle activity specifically did not really influence predator-prey spatial overlap or temporal overlap very much. There weren't too many species that responded to cattle activity. The one exception was this relationship between cougars and white-tailed deer, where we found that cougars were more likely to use areas with increasing cattle activity, but only if deer were also present, white-tailed deer specifically. So this plot here, we have these two different colors. On the bottom of the plot, it shows just the number of cattle detections or images per day. And on the left side of the plot, the y-axis, we have that probability of a cougar using an area based on if a white-tailed deer is also using that area, represented by the pink line, or if there are no white-tailed deer using the area, represented by the blue line. And so with that pink line, we can see that as cattle activity increases, there's this increase in the line, there's this positive relationship. So when there are deer and livestock, cougars are more likely to be in those areas. But if there are no deer, represented by the blue line, cougars are less likely to use these areas where we have cattle, suggesting that cougars are actually avoiding livestock unless their native prey are present. For white-tailed deer, we see different response. So both lines indicate the probability of white-tailed deer using these areas, just depending on whether cougars are present in the yellow or absent in the green. But regardless of whether these cougars are in the area, it seems that white-tailed deer are just more likely to use areas that have increasing cattle activity. So they don't seem to really care whether there's a predator around, they just like to use areas where there's livestock activity. In addition, we found that looking at over the daily cycle, we found that deer tended to increase their activity to being more active during the daytime when cattle were present. And they actually mirrored the times of day when cattle were active. Versus cougars tended to be active at night. So it seems that white-tailed deer were able to actually shift their activity to be active at times when their cattle were active, but the cougars were not, potentially helping them avoid cougar activity over the course of the day. In terms of the hunting season, we actually found that that had a stronger influence, hunter activity in public versus private land, that seemed to have a stronger influence on predator and prey overlap. So first kind of interesting result we found was that deer tended to use private lands during the hunting season, which is probably not a big shock to anyone, but our data also support this idea that deer and other ungulates across the West are trying to use private land more often during the hunting season, presumably to avoid mortality risk from human hunters. Interestingly, we also found that predators tended to use areas that had more hunter activity, which was actually tied more to public lands. So it seems that predators and prey are kind of shifting away from each other during the hunting season and using different areas, which theoretically could change their overlap across space. And in addition, we found that predators were more likely to increase their activity at night during the hunting season, presumably to avoid human hunters, versus deer did not. They were generally crepuscular, active at dawn and dusk, and didn't really change that in areas where there were or were not hunting activity occurring. So with all of that put together, some kind of general takeaways from this study are that one, livestock activity seems to have little influence on the spatial or temporal overlap of most predators and prey. And even when we have exceptions, such as the cougar and white-tailed deer, where we see there's some spatial overlap between these species when there are cattle present, white-tailed deer are still able to avoid interacting with cougars to a certain extent by just shifting the times of day when they're active to times when cougars are less active. We also found that there's this potential for private lands to be really important for deer during the hunting season. So not only does moving to private lands help deer avoid some mortality risk from human hunters, since most hunting activities happen on public land, but it also seems that because natural predators are staying in areas with a lot of human hunters, that this actually allows deer to avoid potential mortality risk from both human hunters and their natural predators. And last, I'll just note that our research suggests that predators might be keying in on some kind of food subsidies that hunters are providing during the hunting season, such as gut piles or wounding loss, which is kind of interesting. These predators seem to be keying in on areas where there is hunting, even though they are also at risk of being hunted themselves. And with that, I will just thank my many collaborators and funders. And for time, I will pass this off to Dr. Ganz.
Thanks so much, Sarah. Just getting set up here. Okay, please chime in if you can't see my screen, but I will get started. Thank you all so much for being here. My name is Taylor Ganz, and my role on the Predator Prey Project is to really look at this system from the perspective of the ungulates. Tonight, I'm going to be talking about how the recent wildfires in the Okanagan have impacted mule deer, and particularly their interactions with predators. So this is a headline from 2018, and it reads, "Washington wildfire season now the worst in history, smoke, haze, impact firefighting." And this is not a surprise to any of us here in Washington. And indeed, wildfires around the world are becoming more frequent, more severe, and larger. Washington state is among the worst impacted areas. And so the causes of these fires are well known. They're driven by human-caused climate change, loss of indigenous burning practices, and fire suppression. But we actually don't have a very good idea about how these fires impact wildlife. Now, mule deer are native to Western North America in these fire-adapted ecosystems, but their populations are largely declining across the West. And while the cause is system dependent, it's largely linked to habitat. And so there's a big question about how these fires are changing habitat and then what the consequences are for mule deer. So to understand how fires are impacting deer, the first consideration is really getting a sense for how fires restructure landscapes. So when a fire comes through, it clears the forest canopy. And in these fire-adapted forests of the American West, this allows more light to reach the understory, and it triggers the regrowth of vegetation. And so what people have observed before is that herbivores may be attracted to these burned areas. So there's a growth of vegetation, but also potentially an accumulation of deadfall. In the winter, though, there's no longer this canopy cover that would otherwise intercept falling snow. And so snow can accumulate to deeper levels in burned forests than unburned forests. So there's a few factors in the ways that this might impact deer. So one is deer mobility is impeded in this deeper snow and burned forests, and their hooves tend to sink in where predator paws function like a snowshoe. And so deer may be more vulnerable to predators in burned areas, but they also might have a harder time accessing forage. So we thought deer might be less likely to use burns in the winter than the summer. The other factor to consider, though, is that these burns also impact predation risk. So where the burn has come through, again, we have this regrowth of shrubby vegetation and accumulation of deadfall. And this can provide really good stalking cover for predators such as cougars that hunt by stalking and ambushing their prey. And on the other hand, wolves are chasing hunters that tend to be most successful in wide open landscapes where they can run. So we might expect that deer would be less likely to use burned areas where cougars were more active, but more likely to use burned areas where wolves were active, using those areas as a refuge. So we had a couple ideas of the possible responses that deer might have to these burns based on these landscape effects. And the first was that forage could be a driving factor, and we would expect that mule deer would be attracted to the high quality forage of recent burns about 20 years post-fire in the summer. But we also thought predation risk could come into play. And so in the summer, we thought that how mule deer responded to burns could reflect the predator hunting mode. And so they'd be more likely to use burned areas where wolves were active, but less likely to use burns where cougars were active. But in the winter, due to that increased vulnerability and deeper snow, we thought that mule deer would just be more likely to avoid burns regardless of which predator was active there. But what we actually thought was most likely was that deer are balancing accessing forage and shifting use depending on predator activity so that they're no more likely to die in a burned area than an unburned area. So to investigate these effects, we are focusing right here in the Okanagan, Methow Valley area. And in fact, if we were in person, we'd be right there down in Winthrop. But what you can see in the blue dots that are moving around is locations of the 150 mule deer that we deployed GPS collars on. Those collars were deployed from 2017 to 2021. And over the course of the year, you can see that deer use quite a wide variety of fire-affected landscapes through their migrations, which are shown in those orange shading for the fires. We also looked at the severity of the fires that had burned, but I'm not showing that here just for clarity. We also needed to understand how predator activity was influencing deer. And to do this, we used GPS data from collared wolves and cougars in the study area. For wolves, we essentially created heat maps of their use with their telemetry data to account for areas where there were known wolf packs and then no wolf pack activity. And those areas are outlined in white here. And for cougars, which you can see in purple, we knew that cougars pretty much fully occupied their landscape. And so we predicted their use based on habitat associations. And so here, darker purple areas have higher likelihood of cougar use, where the lighter areas have a lower likelihood of cougar use. And then we took the mule deer GPS data to characterize which habitats they were using in regards to fire and wolves and cougars. So we start with their GPS data. We have a location every 4 hours. And for each location they use, we generate some random points that they didn't use. And by comparing those locations, we can characterize the strength of selection or avoidance for certain landscape features. So here's what the results look like. I know there's a lot here, but I'll walk through it. So this is just looking at how mule deer are responding to burns. Now, across the top here, we have different time intervals since the fire has burned, looking at 1 year ago or the year of a fire, all the way to 35 years back. And then over on our y-axis, we are looking at the severity of the fire that burned. And for the blue dots, we're showing what deer response in the winter is. And when they're to the left of the dotted line, we see that deer are avoiding burns. So here we see that pretty consistently, regardless of the severity of the fire or how long since it's burned, deer avoid burns in the winter, which we predicted. And in the summer, we generally see that deer are selecting for burns due to that forage enhancing effect, which we also predicted. But how does this change when we then consider cougars in the picture? So now we're going to look at how cougars change this baseline response to burns. So in the winter, we see that where cougar activity was high, deer avoid burns even more, which we expected due to that increased vulnerability in the snow. But in the summer, we see a mixed response. We had predicted that they would be consistently avoiding burns when cougar activity was high, but it really seemed to depend on the characteristics of the burn. And we think that has to do with the specific habitat changes from that fire. Now, if we consider how deer responded to burns with wolf activity, we see that in the winter, deer were a bit more likely to use burned areas where wolf activity was higher. And the same thing was true in the summer. So we really did see that effect where burns were refuge from wolves for deer, with both increased forage and lower predation risk. The other thing we looked at is the different causes of mortality for these deer. And we wanted to understand if their behavioral response to burns was in any way increasing their vulnerability to predation risk, but we didn't find any indication that deer were more likely to die in a burned area. Now, cougars were the primary predator of deer, followed by humans, although I should note that there were quite a few unknown causes of death. But no matter what we looked at, there didn't seem to be an effect where fires were directly increasing predation risk or mortality risk. So just to recap and sum this all up, we found that deer generally selected for burns in the summer, but were avoiding them in the winter. And predator traits influenced how they use these burns in both seasons. So there was some evidence that there might be this trade-off with cougars where deer had to balance accessing high quality forage, but potentially with increased predation risk. But it was really a win-win for burns as far as wolves were concerned. And we again saw that deer were balancing this predation risk with accessing forage. So what does this all mean? Well, in predicting the impacts of burns and other systems, we really need to consider both the changes to the landscape structure and the predator community that's present. So we may see a very different response in a system that's very wolf dominated versus one that's cougar dominated. And there are trade-offs between enhanced summer forage, but if deer are avoiding these burns in the winter, that could be a reduced winter range. And we don't really know which one of these wins out. I'd be interested to look into that though as a next step. But thinking about other systems, it may have to do with the latitude, the climate, and the elevation, shifting the balance of these costs versus benefits, dictating forage quality, and then also how much snow is on that landscape. So with that, I just really like to thank all of you for being here, the Wenatchee River Institute and the Methow Conservancy for hosting us, and the many people who contributed to this project. And from there, I'm going to hand it over to Lauren.
Thank you so much, Taylor. All right. Okay, so hello everybody. So yeah, my name is Laura Satterfield, and I looked at the diets of wolves and cougars in this multi-prey human-dominated system that we have here in Eastern Washington. And kind of going back to some of the things Sarah was talking about, we have top predators that are returning to many regions of the Pacific Northwest and also different parts of the globe. And these landscapes that have multiple different predators, they influence prey population in different ways, as Taylor just discussed. But there are fewer studies of these predator-predator dynamics in managed landscapes. So by managed landscapes, we mean areas where we have, timber harvest and recreation, and the landscape isn't specifically protected for nature or wildlife, although parts of it may be. And we have fewer studies from multi-prey landscapes, so landscapes that have multiple different primary prey. So here in the Okanagan, we mostly have mule deer, but in our other study area, we have white-tailed deer and elk as well. Okay, so when we talk about top predators, specifically here in Eastern Washington, we're talking about wolves recolonizing after an 80-year absence. So wolves were historically occurred across the Western US and were killed off in the 1930s through a combination of local and federal persecution campaigns. But they came back to Washington in 2008. And in fact, the first pack discovered in Washington was the Lookout Pack right here in the Methow Valley. And so the prey that we were looking at for wolves was mule deer here in the Okanagan, but also white-tailed deer, elk, and moose. Cougars, on the other hand, have been in these areas for the whole time, including the period without wolves. And so these cougars now are having to adapt to the renewed presence of wolves. And we wanted to see how that was going to affect the diet of both of these predators and also how much those diets overlap. So our main goal was to ask how much do wolf and cougar diets overlap? How does prey diversity affect dietary overlap? So there's a kind of theory that if you have more prey species, so prey diversity, that there's more opportunity for animals to separate their diets. So we were kind of predicting when we have more prey species that predators have more opportunity to eat different things. And we looked at 2 different types of diet measures so that we kind of had 2 lenses to look through. One was scat. So this was, poop that we collected by tracking collared wolves and cougars. And we also went to GPS clusters like you see here, which is basically a place where we got a bunch of GPS points. And that meant the animal had kind of been hanging out there. So either they were resting or sometimes they were eating something. So just as a reminder, the Northeast study area is over here, over on the border of Idaho, and really has a denser number of wolf packs. Whereas here in Okanagan County, when we were starting the study, we actually only had the Lookout and Loop Loop pack. And Navarre and Sullivan Creek have come in towards the end of my sampling period. Okay, so what did we predict? First, I predicted that dietary partitioning would be greater in landscapes with more diverse prey. So basically in the Okanagan, we only have, we have mule deer and whitetail deer, but mule deer are predominant. Whereas in the Northeast, whitetail, elk, and moose are all fairly present prey species. So because of that, I predicted that in the Northeast study area, wolves and cougars would be able to have diets that were more different than in the Okanagan. And similarly, I predicted that in seasons with more diverse prey, we would also see more divergence of diet. So in the summertime, there's a lot more animals out on the landscape. You've got a lot of animals that were hibernating over the winter coming out, small and medium-sized mammals, birds, these sorts of things migrating back. And so that provides an increased number of prey species available during the summer. And so over the course of the project, through combined efforts of the University of Washington and the Washington Department of Fish and Wildlife, we were able to collar 60 cougars and 16 wolves and collect scat by following these individuals around to their cluster sites. So when we would get there, we would identify the prey at the cluster by looking at morphological features, and we would also age and sex the prey, although I'm not going to go into those results here tonight. What I'm going to focus on tonight is really the scat analysis. And what was really exciting was that we were able to use genetic methods to analyze the scat rather than using the conventional method where you essentially wash the scat and you're left with hair and bones, and you use those hair and bones to identify what prey were eaten. That is a really labor-intensive and time-consuming method, and it's really hard to identify rare prey species because there might just be, a few hairs from a chipmunk or something. Whereas using genetic methods, we're able to more reliably detect those rarer species and get a better idea of prey use across the animal's diet. So in order to do this, we had to spend a lot of time in the lab tubing scats. That's what you see this student doing here. So we had a number of undergraduate students that worked for us during COVID, which is a very, very interesting experience. In fact, all of us had to write up protocols during COVID. And so we spent a lot of time tubing scats. You'll see now in the bottom right slide here, this is kind of what ultimately got sent to the lab. And it represents hours and hours of work, hundreds of hours of work when you consider the field work too. So the genetic process we use is metabarcoding, and this is a really cool method. So traditionally with genetics, it's, or at least what was usually done pre-meta barcoding was that you would say, okay, I want to know if there's moose in this scat. So you're going to test specifically for moose. And then if you also want to test for white-tailed deer, you have to come up with a different set of what they call primers, but basically like a different kind of solution that's looking for those genetic markers of whatever it is, moose, elk, etc. With metabarcoding, we can test for the entire vertebrate genome at once. So what it does is it looks at a section of mitochondrial DNA, and basically this section is available in all vertebrates. And in that way, we're able to identify at least down to species, or sorry, at least down to genus, a lot of the species in the diet. And it's very sensitive, so we can detect rare species in addition to the common ones. It's also that sensitivity means that we can use scats that are quote unquote old. Maybe they've been out on the landscape for months, been rained on, frozen, thawed, etc. But this method is very sensitive, and so it can still detect DNA in a lot of cases. And also conveniently, because it tests all the DNA in the scat, it will also tell us who the depositor likely was. Because, for example, if it's a wolf scat, you're going to get a lot of wolf DNA in addition to whatever DNA of what the wolf ate. So we were able to confirm that our scats were actually from the species that we identified them being from in the field. So yeah, lots of time spent in the lab picking these scats apart, and lots of undergrads that I'm very grateful to. And so this is kind of the result. We collected scats from we collected about 400 scats and submitted them for analysis, and approximately 150 wolf and 150 cougar scats came back and were able to be used because they amplified and they had prey. Okay, so what did we find? Looking at both the cluster and the scat, we kind of compared these two methods to see what each detected. As far as cougar diets, both clusters and scat showed cougars eating elk, moose, mule deer, and white-tailed deer. Not super surprising, but very cool that, we confirmed that. Also, both methods allowed us to detect beaver, raccoon, coyote, snowshoe hare, grouse, turkey, small mammals, and some unknown birds and canids that we were only really able to get down to genus. With the clusters, however, we were able to find that cougars were actually also eating cougars. We saw cougars eating livestock and porcupines, whereas from the scat, we saw bobcat, domestic cat, shrew, and squirrel, all in very low percentages. For wolves, again, we found elk, moose, mule deer, white-tailed deer, and we also found grouse, snowshoe hare, turkey, and livestock. Clusters allowed us to find that a lot of the livestock detected was actually from carcass dump sites. We found really only carcass dump sites. We didn't find any cases of wolf predation on livestock that the wolf actually killed. It was all scavenging. That doesn't mean that didn't happen. It's just not something we detected. And we definitely found a lot of dump sites that wolves were very aware of and revisiting repeatedly. In some cases, traveling over 10 miles to go to the dump site and back. Magpies, marmots, skunks, beavers. We actually found wolves eating black bear in 5 scats. It's again, unsure if that was like eating gut piles, as Sarah had mentioned, or whether they were actually killing these bears. We don't know, but we found it in their scat, along with bobcat, chipmunk, fish, flying squirrel, etc. Okay, I think I'm low on time, so I'm gonna try to speed this up. But so what did we find? We had results from the Northeast and Okanagan study area. So Northeast are the left 2 columns in all of the following plots. Okanagan are the right 2 columns in all the following plots. And within each plot, the left side is summer and the right side is winter. So we have summer here and winter here for cougars in the Northeast, for example. So first I'm going to look at the Northeast study area. This is the study area that had a lot more prey species. Specifically, it had a lot of white-tailed deer and moose, in addition to elk and mule deer. What we saw was that cougars in the Northeast study area were eating predominantly white-tailed deer, but they were also feeding on moose, particularly in the summertime. And you see in the summer this wide array of other species found. So elk, birds, carnivores, lagomorphs, so like snowshoe hare, etc. And less in the winter. This is following our prediction that they're going to use more prey species when they're available. Similarly, in the for wolves in the Northeast, we also found them eating large amounts of white-tailed deer and moose. And actually, that there being kind of a switch where in the wintertime, more whitetail was used than moose, and the opposite in the summer. Oh, not advancing. Okay. For elk, we really found minimal use of elk. Mule deer, minimal use of mule deer. Whereas in the Okanagan, again, as we predicted, all these green bars are mule deer. This is the prominent prey here. It's not surprising that so many of these scats contained that prey species. So I forgot to say, though, the left side here, 0 to 1, this is kind of the percent of scats that had this prey species. So here about 75% of scats had mule deer, and here it's a little bit over 75. And you see for wolves in the Okanagan in the winter, it's close to 100% use of mule deer. We do know now that they're also eating moose. We just didn't detect it during our study, and that could indicate changes in the moose population, or maybe we just missed it, but certainly not a huge part of their diet when we were doing this work. And again, you see across the board this increased use of multi-prey species in the summer. Finally, livestock played a pretty big role in Okanagan wolf diets in the summertime, and again, and also in the Northeast. And again, we found that the majority of this was scavenging. Okay, so at the end of the day, we did find that predator diets overlapped less in areas and seasons with more prey diversity, so in the Northeast study area and in the summer, and that moose, white-tailed deer, and livestock all supplement wolf and cougar diets seasonally and allow for divergence of diets. All right, and with that, I'm going to thank the other people on the Predator Prey Project team, my fellow grad students and PIs and agency folks, and also a huge shout out to landowners who allowed us access to their private land to do this work. You all were absolutely instrumental in our ability to actually accurately characterize all of our work, including diets. And so thank you to those in the audience today. All right, and I'll pass off to Laura Prugh.
All right, thank you, Lauren. All right, let me get my screen sharing going. Okay, hopefully you're seeing the title slide there. I was heading up the mesopredator side of this project, so looking at how medium-sized predators, known as mesopredators, are affected by wolves and also cougars, so focusing on coyotes and bobcats. And we were conducting the study in both study areas, but today I'm going to focus on our findings from the Methow specifically. And I would like to acknowledge my postdoc, Callum Cunningham, who's been very helpful in analyzing these data. And also, just to note, this part of the project was not funded by DFW. It was funded by the National Science Foundation, so it was kind of an add-on to the Predator Prey project. Okay, see if I can get my slides to advance. Okay, so I want to just start by giving a big shout out to Becca, Carmen, and Anna, who were extremely instrumental in the field work. And strangely enough, I don't have a photo of all three of them together, so I kind of stitched these two photos. So they met working on this project. Carmen and Anna worked on the project for all 6 field seasons, and Becca for nearly the whole project. And they met while working here, so that the spawning of their
nonprofit home range research is, I think, one of the great products of the Methow project. So one of the things that we were interested in looking at was this, whether this phenomenon that's known as mesopredator release might be reversed with the return of wolves. So this is the idea that large carnivores can keep populations of smaller predators in check. And it's certainly possible that when wolves were extirpated from Washington in the 1900s, that populations of mesopredators like coyotes increased in numbers. So that's this phenomenon known as mesopredator release that my research lab has been looking at for some time. Let me try to get rid of that. Okay. So this phenomenon of mesopredator release or mesopredator suppression has been shown in protected areas, but in areas like the Methow or northeastern Washington, where there's a lot of human activity, that might really limit the distribution and activity of cougars and wolves. So whether they can still suppress mesopredator populations is kind of an unknown that we wanted to look at. So we had a few research questions. Number one, just what are the rates and causes of death for coyotes and bobcats? Second, what are their population sizes? Third, what are they eating? And I grade this one out. How much are they scavenging? Because that's a component of the project that I won't be presenting today, but it is something that we were examining. So we used three main methods. One, radio collaring to look at their movements and mortality. We also collected a lot of scats, and this was to look at their diet and also to estimate their population size using fecal genotyping. I think a lot of the volunteer scat collectors, as part of the program that Becca put together, might be here in the audience. So thank you all. There were 40 dedicated volunteers that collected scats, and that was really helpful. And we also deployed cameras at ungulate carcasses to monitor scavenging. So with the, let me see if this will play. For the collaring, we would, for bobcats, we'd put out these cage traps and put little cat toy out there, and this bobcat did eventually go in the trap after playing with the cat toy for a little while. For coyotes, they are much more wily, as is, their name implies. Wily coyotes really are wily. So we had to use padded foothold traps to catch them, because there's no way they'd go anywhere near a trap set like this. Let me, move on here. So straight to the results from the collar data. So in the Methow, we collared 19 bobcats and 19 coyotes, and these are their fates. So we had one bobcat and one coyote that were each killed by cougars. We also had two coyotes that were killed by other coyotes, and that was where the genetics work came in handy, where we swabbed bite wounds. Because initially from the field assessment, it looked like they had been killed by wolves, but it turned out they were killed by other coyotes. We weren't able to identify the predator. We could tell they died of predation, but weren't sure which predator for one bobcat and coyote. Humans was by far the biggest source of mortality for both bobcats and coyotes. We also had in this other category, one bobcat drowned, fell into a river in the winter and couldn't climb its way back out. And one either died of disease or starvation. 2 coyotes, we weren't sure how they died. And then several bobcats and coyotes survived to the end of the project. So overall, the coyotes had about a 55% annual survival rate and about 60% for bobcats, which is fairly typical for these species. Interestingly, we had no documented wolf-caused mortalities of our collared animals, although there were, these are some coyote pups that were found at a loop-loop wolf cluster. And when Lauren's crew and our crew went to clusters, there were other coyotes found at cougar clusters as well. So we knew they did occasionally get killed, but our collared coyotes were not killed by wolves. So when we estimated their mortality rates from these different causes and kind of lumped categories together, and this is looking across both of those study areas, about 1 out of every 4 bobcats is killed by people every year, and versus, so that's this bobcats over here, human mortality rate is in the orange. And then for large carnivores, which across both study areas was cougars and bears, only about 7% mortality. And then this other category, disease, drowning, and intra-specific fighting. For coyotes, 31%, so almost 1 out of every 3 coyotes is killed by people every year, versus about 10% by large carnivores. So in these landscapes, the risk of mortality from people is about 3 to 4 times higher than it was from large carnivores. Looking at the collar data in terms of their movements, there were a few things that were notable. For one thing, bobcats had gigantic home range sizes in the Methow. So their home ranges, the median size was about 64 square miles. And there was a very large range among individuals. So the smallest territory was 26 square miles and the largest was 255 square miles. So really huge. Whereas coyote ranges were about 4 times smaller than bobcats. And in the Northeast, the range sizes were all around, for bobcats and coyotes, were very similar to the 16 square mile size. So the real outlier is the bobcat territory size in the Methow. And that's a big mystery for us. I'm very curious if any of you have hypotheses about why that would be. So you can think about that, maybe put some ideas into the chat. But moving on to the diet results. So here we collected over 1,500 scats total in the Methow. 230 were from the Methow Conservancy volunteers. And from these scats, we were able to identify 254 unique individual coyotes. So we collected between 1 and 22 scats per coyote. And so just wanted to note, we did identify all of our radio collared coyotes through our scat sample. This map shows in red the routes of our regular scat routes, and then the dots are routes from the Methow Conservancy volunteers. So that was 13 times more individuals that we were able to identify with the non-invasive scat collection method compared to the much more intensive and invasive method of radio collaring the collars, the coyotes. All right, so what were they eating? Their diet was very diverse. We looked at diet just for the coyotes. There were 18 mammal species detected, and there was a lot of plant in their diet as well. About 90% of the diet was made up of 5 items, so ungulates, and a lot of that seemed to be from scavenging in the winter. There was some fawn predation in the summer as well. Small mammals, and then seeds and berries was a big part of the diet, as well as other plant matter. And then bunnies was the 5th most important item. And this was work by an undergraduate who did her senior thesis examining the coyote scats. So just looking more specifically in the Methow, so that's the white bars here, there was less ungulate in the diet compared to the Northeast, and less small mammal in the diet, and a lot more seeds and berries. So when we combine the seeds and berry category with the plant matter category, plants were about 45% of the coyote diet in the Methow, and that's looking at winter and summer. So in the summer, their diet has a lot of vegetation in it. Okay, so just to summarize, humans were the most important cause of mortality for coyotes and bobcats. Bobcats have huge territories in the Methow, and we don't know why. And coyotes may be functioning as important seed dispersers in the Methow Valley. So I also want to give a big thanks to everybody who returned radio collars to us, who allowed us to come onto your land, to those 40 Methow Conservancy volunteers, to Lauren and her crew who collected some scats and was collaborating on the project, and then to our crew, and I just put our Methow crew here. We also had a crew in the Northeast, but there were 96 other project participants, undergraduates, and volunteers who took part in the project. So it was a really huge effort. All right, so let me stop sharing now.
Thanks so much, y'all. We have some good questions coming in, and I think some of them are for specific people, but I think, you know, unless it's for a specific person, feel free to leap in with whoever, whoever's most interested in answering or most prepared to do so. Let's start with, there's this question from David who says, would you assume that environments with a more complete suite of predators would have healthier prey populations? Would the presence of this suite of predators protect against the likelihood of chronic wasting disease, for example?
I think that one was directed at Lauren, or it came in during Lauren's talk. So I don't know if you want to field that one, Lauren.
I'm so sorry. My internet cut out. Could you repeat that question?
Yeah, and I think you can see it in the chat too if we cut out again, but it says, would you assume that environments with a more complete suite of predators would have healthier prey populations? Would the presence of this suite of predators protect against the likelihood of chronic wasting disease, for example?
Oh, yeah. So that's a tough one. You know, we weren't really looking at body condition of prey, because when we would get to these sites, you know, often all that was left was bones. And, you know, you can tell something about body condition from bone marrow content in femurs, but usually that's kind of like once they lose bone marrow fat in their femur, they're really like severely starving. So it's hard to pick that out the way we did it. However, you know, research does tend to indicate that like wolves as coursing predators that chase their prey, they do tend to kill the older and younger and or weaker of the prey species. And our clusters did show this. Whereas cougars are more likely to kind of, they do still key in on animals that are a little bit easier to kill. So like fawns, for example, everybody likes fawns. As far as chronic wasting disease goes, though, yeah, this is a little bit doomsday, but nothing's going to stop chronic wasting disease, you know, unless we as like a human community really put effort into keeping bones and like contaminated animal parts from crossing state lines. Chronic wasting disease spreads when deer congregate. So like when people feed deer, you know, deer congregating, that can spread chronic wasting disease and so forth. So I wish that wolves and cougars could be the answer for chronic wasting, but I think the answer is really going to be us as humans. Yeah. Thank you for that question.
Well, I can ask the next question. So Adrian asks, in Eastern Washington, residents and hunters claim that deer and elk populations have significantly decreased recently. And they attribute this to predators, specifically cougars and wolves. Did your research find a significant population decrease in deer and elk?
I'm happy to field that question. So as far as mule deer went, which I just talked about and which was our focus in the Okanagan, unfortunately, we weren't able to look at an overall population trajectory because we were only tracking adult females. However, in our northeastern study area, we did look at population trends for the white-tailed deer and the elk. And I just want to start by saying, I think one thing that's challenging is our study was for 4 years, 2017 to 2021. So we have a sense of what the population was doing at that time, but not in reference to what the population may have been 20 years ago or longer. That said, we actually found that the elk population is increasing, and survival of adult females in particular is quite high. So the elk population was increasing, so we didn't see a decrease there. The white-tailed deer population, statistically, our estimates are that the population is stable. It may have been somewhat decreasing, a small amount, but that was a statistically non-significant result. However, in terms of the factors that were driving that population, one factor that was definitely contributing to population growth was cougars and wolves. But the other really important factor that we looked into was forage dynamics in the system. So both the availability of food for white-tailed deer and those predators are influencing their population trajectory, which during our 4-year period was stable to maybe declining a little bit. In addition to just recognizing that our study was only a 4-year period snapshot in time, it's also important to consider that the hemorrhagic disease outbreak that happened in 2021 occurred right after our study concluded. And so we weren't accounting for potential population impacts of that hit the Northeast area pretty hard.
So we have several questions that I think are getting at the same thing more or less, and that is lots of questions about human-caused mortality of the mesocarnivores. And so, yeah, can you speak to sort of what those causes were? Do we know a lot about, the legality of the deaths and the cause of those deaths?
Sure, yeah. Nearly all of the bobcats were trapped, and that was it was pretty much all males that were trapped. So the bobcat trappers tend to release females when they catch them. There were 2 bobcats, I believe 2 or 3 bobcats that were legally shot. And the coyotes were all shot for various reasons. There was one bobcat that was shot by a landowner after killing 17 chickens. Chickens seem to play a role in the mesocarnivore deaths. There was one of our collared coyotes that I went to the mortality investigation was shot by a landowner when it was trying to get their chickens. So livestock conflict was a major driver, I think, especially of the coyote deaths, but also some of the bobcats.
All right, I have a question here. I think this might be for Lauren. What were the most prevalent plant and berry species in the coyote diet?
Oh, I think that's one for Laura Prugh.
Oh, I'm sorry, Laura.
So I am not sure that, so Anna, the undergraduate who's doing that thesis, she was using traditional diet analysis where you wash the scat and Sage, can you stop scratching yourself? My dog is scratching his collar and it's really loud. She was doing the traditional kind of scat analysis where you wash the scat and then look at the remains and compare it to a reference collection. So not the super awesome technique that Lauren's wolf and cougar scats with the metabarcoding. We are using that on other projects now, and it's really great. But so I think she was just identifying the berries as like berries and not didn't have the expertise to identify them to species. But I do remember seeing some coyote scats that looked like they had knick berries in them and some that look like blueberry scats. But we don't really have good quantitative data on that.
So we have a question about links and whether links were involved in this project specifically at all. I know, just a quick shout out, I guess, looking toward the future, we do have a couple events slated for this year with the Home Range Wildlife Crew, who has already gotten their shout out for the night, but they're going to be talking about their active links project in September as a totally separate event. But I think the question still stands: were links involved in this first round?
No, they weren't. We were just focusing on coyotes and bobcats. So we weren't trying to trap links or include them as part of this project. I guess Sarah would be the main one who has information about links from the cameras.
Yeah, I was just going to chime in real quick. We did detect links on cameras in the Okanagan study area, particularly kind of like north and west of Winthrop. But one, they weren't a primary focal species. A lot of times where we put cameras, we're probably a little too low of elevation to get good data on them. And then also because of that, we just had too few detections of links to really incorporate them into our analyses. So we have a little bit of data, but not enough to really do anything with it in terms of the camera data.
I had a question about, I guess, the livestock component of what you all talked about. And I was curious, so it sounded like most of the livestock that was being found in scat was coming from these dump sites. Is there any reason that those dump sites are a bad thing ultimately? Like, is that something that's maybe going to lead these predators to actually want to feed on livestock, or is that like a silly horror movie trope that they're going to get a taste for cattle and then, maybe change their ways?
Thanks for that question. That's a really good question. Again, we weren't researching that specifically, but it's very unlikely that like eating cow meat would like cause an animal to be like, okay, now I'm going to go chase down this live thing, like animals need to kind of get used to a certain animal as a prey item. And cougars from our research really, if they're going to take livestock, it tends to be sheep or goats or something smaller, rather than cattle. And for wolves, certainly wolves have been recorded in Washington, killing adult cattle and that kind of thing. But, again, we didn't find that. I think as far as the dump sites go, they're certainly not good. I'll put it that way, because often these dump sites are where people are needing to dump carcasses. And so where is that going to be? It's going to be near like a dairy farm or some other operation where there's live animals. And so I think that it's more that it would attract, whatever predators to an area with live animals and have them coming there repeatedly. That would be a risk, because then at some point, one day there's going to be an animal that's limping or an animal that, just gave birth or whatever. And then you might be having an issue at that point. And so I guess, sorry, to close that out, one approach that's been discussed in the past is fencing off these dump sites or having community dump sites. But these are really hard, because often, people will have a dump site on their own property. And so there may be many of these out there. But yeah, they do tend to draw predators closer to people.
I have a question here. Is there any indication from meso carnivores being more abundant or using the landscape differently in areas of wolf concentrations? What has been the impact of wolves on meso carnivores?
Sure, yeah, we're our next phase of analysis is to take those data from the 253 coyotes that we identified in the Methow, and then whatever that number is in the Northeast, and model the density of coyotes on the landscape in relation to the wolf territories. We expect, based on looking at their movement patterns from the radio collar data, which we have looked at, I predict that there will be higher density of coyotes outside of wolf territories compared to within, because they strongly avoided wolf territories, the movements of when we looked at the movements of our radio collared coyotes. The bobcats did not avoid areas where we had collared wolves or cougars to the extent that coyotes did. Coyotes avoided wolves very strongly, and then cougars less strongly, but still avoided them. So it seems to be mainly, I think the wolves and coyotes are limiting their activity and possibly abundance in these landscapes.
So I think for the sake of time, unfortunately, we're not going to get to all the questions, but I do want to maybe close out on this one. Someone messaged me asking essentially what lingers now, like what's the burning question in your mind about what's next? And I think, Laura, what you just talked about is maybe one of those, but I would love to hear from everyone what's kind of the one thing that you're still, have a burning curiosity about.
Well, I guess since I'm unmuted, I'll kick it off. Yeah, so in addition to looking at the density dynamics, we're in the process of looking at the scavenging dynamics as well, and kind of putting those together to see what are the patterns of mortality and scavenging, and what's the net effect on the density of these mesocarnivores.
I'm happy to jump in. So one of the things that I shared with you is that we see this effect where mule deer in the Okanagan are using burned areas in the summer. They're keying into that, we think because of the improved forage, but they're avoiding those burns in the winter, which kind of as a functional form of habitat loss if they're not using those areas. And we found that they weren't any more likely to die in a burn than outside of a burn. So there's no direct effect of these burns on mortality by predation risk. But I would be really interested to know if, over the course of a year, if they're using these burns, if that influences their survival. For instance, if they might be in better body condition due to that forage enhancing effect, or because of that limited habitat in the winter in poorer condition. So that's something I would be eager to look at more.
So with the research I presented tonight, a lot of it is basically just based on patterns of, this is where we're seeing animals, activity patterns, but we don't really know based on the camera data what the ultimate consequences of those patterns are. So if we're seeing shifts in overlap of predators and prey, either spatially or temporally with in response to either cattle or hunters or land ownership, we don't really know if that actually then equates to changes in predation risk or changes in mortality for these different species. So I think a really interesting future avenue to go down is to then, based on the data we found, or data we have in those relationships, see if we see ultimate consequences using, hopefully, some of Taylor or Lauren's data looking at mortality and survival of these different species, since we have some of that data collected by this project. So I think that would be a really neat future avenue for my particular research, is to see what do these patterns ultimately result in for these different species.
Yeah, just kind of going off of what Laura, Taylor, and Sarah said, for my research right now, I'm looking at diet and dietary overlap, but we're also going to use the GPS collar data from the wolves and cougars to look at some spatial dynamics and temporal dynamics from the camera traps that Sarah deployed. So the next step is, for at least from my kind of section of this, is to look at where wolves and cougars are on the landscape, and whether or not their overlap is mediated by human presence. So like, is human presence having some effect on when and where wolves and cougars overlap? One thing that came out of the diet data was that cougars are eating white-tailed deer in the Okanagan, whereas wolves are not. And those white-tailed deer are primarily, down in the valley bottoms where people are, where houses are, really where the rivers are as well. And traditionally you would expect wolves to be down there at the valley bottom, but they're not, at least not right now. And so it begs the question whether there's kind of a human shielding effect going on, or, that wolves are just less tolerant of people than cougars. So we're, still looking at these dynamics, but that's one thing that is kind of in the works right now. Also, the next thing too is to kind of look at, okay, even if wolves and cougars are overlapping, are they making their kills in the same places or not? Or are they spatially separating where they're making kills? So that'll give a sense of whether there's kind of duplicate pressure on prey populations from both wolves and cougars in the same area or not. My prediction there really is that everyone has to go where there's food. You got to go to the grocery store one way or another. And so just from looking at the data, it appears that wolves and cougars tend to go where there's more prey, but that they, may be interacting with people differently. And then finally, we had camera traps on cougar kills because we were trying to understand, initially, if other predators were coming in and taking food from cougars. We found that wolves generally weren't, or if they did come, it'd be a scavenging event, but that cougars did have a lot of food taken from them by black bears. And so one thing I'd also like to do going forward would be look at the role of black bears. And we actually have data on black bear predation on elk and calves through Taylor's project. We have black bear distribution data through Sarah's cameras and also from some WDFW surveys. And then we have this data from the camera trap showing often black bears and cougars, alternating feeding. on a carcass. So yeah, trying to get bears in the dynamic is something else that would be really interesting, especially with all the discussion about grizzlies coming back. It's important to kind of understand how all these predators are interacting with each other in terms of diet.
Plenty of work to do. Well, thank you all so much for giving your time tonight, and so cool that all this is happening kind of in our backyards here in the Methow, and looking forward to learning more about the next steps. And thanks to everyone who joined to learn alongside me tonight, and hope to see you next month at our first Tuesday at the barn.
All right, thanks everyone.
Thank you.
Thanks, everybody.
Thank you.
Thanks so much for having us.